All right, so now we're actually going to get into kind of how the contraction actually occurs, which is really focused on one concept, and that is the sliding filament theory, or you may also see it called the sliding filament model. We're going to talk about that whole cycle, that whole process. So the last video kind of got us up to speed in terms of the moving parts, and then we're going to get into how we actually cycle through that, through contraction and relaxation. So remember that a muscle cell or a muscle fiber is composed of all those myofibrils, which are made up of those filaments, a mix of filaments like actin and myosin. And so we see like, it's like almost like we've packed up a bunch of spaghetti noodles. And we have this repeating pattern of these sarcomeres, and that's the striations. That's where those striations are coming from. We're surrounded by the sarcoplasm around each of those, right? And then we have the sarcolemma all the way around, right? The new basically name for the plasma membrane here. And so we have a few different components of the sarcomere. We mentioned sarcomere in the last video, but we didn't really talk about what it is. But it is the functional microscopic unit of muscle, right? The sarcomere is basically the component that is going to contract. And as all of these sarcomeres contract together, that is what's going to lead us to contract the entire muscle. So we have a few different regions, a few different like spaces and terms used to organize the sarcomere. Number one is the A band, right? The A band is this right here, from here to here. This is going to be slightly darker if we were to look with like at a micrograph. This is where we have the thick filaments and some thin filaments. So for the most part, we're talking about the A band is where like the myosin resides. So we see it like kind of runs the entire length of the myosin, and some of that actin overlaps. The I band is over here, and it's the thin little one, and that's only where we find actin. The Z disc passes through the center of the I band, and it kind of, in a way, creates the boundary from one sarcomere to the next. So a sarcomere is going to run from one Z disc or Z band to the other, right on the other side. The H zone right here is where we only see the myosin, the thick filaments, and the M line is going to be the myomesin, where that myosin is anchored down. So altogether, these components form what's called the sarcomere. Again, is the functional unit of muscle, right? Is the microscopic functional unit. And so if we were to kind of peel this away and kind of look at what components are making up what, here is our sarcomere. Here's our Z disc we see. Here is our myosin. Here is our actin in yellow. The M line that the myosin is linked to. We see the A band runs the entire length of the myosin, including the overlap of the actin. The I band is where we just find the actin, right? The H zone is where we just find the myosin. And so now we get into the sliding filament theory, or the sliding filament model, or sliding filament mechanism. I've seen a bunch of different names for this. We're talking about the same thing though. And that is that myosin will pull on actin, causing the thin filaments on each side to slide in towards the middle. So basically, the myosin doesn't really move. It's pulling things in towards the middle of itself. So this Z disc here and this Z disc here will both move in towards the middle, or I'm sorry, this one here and this one here will both move in towards the middle, right? And then this Z disc and this Z disc here will be pulled in towards the middle here. So we see like all together, right, this gets pulled in. This is partially contracted. Here it's fully contracted. We see we have gotten much, much shorter, and that we have significant overlap of all those different filaments compared to when we started, where there was very little overlap there. All right, so let's go ahead and look at what's going on here, because this is really important for you to be able to talk about. So I'm going to start with number one and then cycle around, because you could really start anywhere, but it's easiest to start there. And so we take ATP, we break ATP down into ADP and phosphate to recock, basically reset the myosin head. Then we can bind to actin to form the cross bridge. When we do that, we then need to conduct the power stroke. And the power stroke is going to come from when we lose the phosphate and the ADP. When we're empty, we don't have anything attached, we perform the power stroke. And then fresh ATP is going to be what allows us to release that actin. And so we're constantly going through this cycle of fresh ATP to let go, break ATP down to reset, lose the ADP and phosphate to perform the power stroke, fresh ATP to let go, break ATP down to reset, lose the ADP and phosphate to perform the power stroke, fresh ATP to let go. And we're doing this process over and over and over and over again. All right, and this is just kind of demonstrating that same thing, so I'm not going to spend much time here, but if you want to read through it, you totally can, right? But again, it's just that big thing of the three main things that are happening, which is we are grabbing onto actin, we're performing a power stroke, we're letting go, we're resetting, right? Over and over and over again. But the big thing is that each of these power strokes, kind of like rowing, is that one single power stroke is not going to cause the muscle to fully, or that sarcomere to fully contract. We go through this a few times, kind of like if you were to row a boat, right? You want to get to a certain distance, you have to row multiple times. And so we're going to perform several power strokes to kind of climb our way in to shorten that sarcomere. Now, in order for this to happen, we need calcium, right? Calcium is required for this because if there's no calcium, the tropomyosin is blocking the binding site, and so we can never do the power stroke then. But if we increase the calcium concentration, we can start that contraction and continue it or sustain it. If we decrease the calcium, this will slow it down or stop it. And again, that's because calcium is needed for that tropomyosin to move, right? When the calcium binds to troponin, then tropomyosin will move from those binding sites. The the sarcoplasmic reticulum will take that calcium back to stop the contraction. So we're just kind of letting calcium get over to those to the sarcomeres, and then also removing it to help control when the muscle is contracting. As long as the sarcoplasmic reticulum leaves calcium out and about, then the muscle will contract. But then when we want to stop contracting, we bring that calcium back into the sarcoplasmic reticulum to stop it. And when calcium levels drop, we don't have any calcium left to really bind to the troponin, which means those tropomyosin blocks the binding sites again.